Right, so here's a video about the proteome and what does it mean? What is that word? And we're also going to give us another word in this section. We're going to talk about something called the genome. So when we think about this, we want to go, how many different proteins are there? We looked at protein synthesis and we looked at things like alternative splicing. We've looked at the different ways that they fold throughout protein hierarchy. And so it kind of leaves the question, how many different combinations are there? And that is what the proteome is, right? So the proteome is simply all of the different proteins within an organism. So in the human, for example, we've probably got almost 1 million, definitely over 200,000. We're not even sure how many proteins are in our bodies, but definitely there's a whole heap of different proteins. So we've got heaps and heaps of different proteins. And then we've also got this other word here called a genome. So a genome is the complete set of genes or genetic material that's present in a cell or an organism. And this tends to be a little bit smaller. And what I mean by this is where we have a look here. So this is within a human. So this is human genome and human proteome. So you can see down the bottom here, the genome is made up of anywhere between 20 to 25,000 different genes. Now, I know that I've said before that there are 3 billion base pairs, but when you chunk that up into chromosomes and then you chunk up the chromosomes into genes, plus then you've got extra sections that don't code for anything, you end up with from 3 billion base pairs, we come down to about 20 to 20,000 genes. It's not necessary that you remember that exact number, but I want you to remember a comparison between the number of base pairs and the number of genes. So heaps and heaps of base pairs, not as many genes, because it takes quite a few base pairs to make up one gene. And then what happens is you get the process of transcription, which is what we've talked about here, and also RNA processing. So this here is after you go through transcription and RNA processing, what happens is we've got a few different ways, like there might be promoters in different parts of the chromosome, which would start a different gene kind of transcribing. Then you've got alternative splicing. You've also got mRNA editing. So sometimes they even delete other parts of the mRNA. And what we end up with is from this 20 to 25,000 genes, we end up with a bit more variation and we go to about 100,000 different types of transcripts. Yeah. And what I mean by transcripts is there's more different types of mRNA than there are types of DNA. And that's because of all these editing processes that we go through. Now, after you've done that, we have to go through translation. And when you go through translation, what you end up with is even more variation. So now your sequence of amino acids that's been created, so this is translation and post-translation, they can fold into different alpha helices or alpha helices in different places, beta pleated sheets in different places. Then depending on how the 3D structure forms in its tertiary structure, all of this ends up with about over 1 million different types of proteins. So you've got genome is a lot smaller than your proteome. Again, don't need to remember those numbers, but you just need to know the genome is smaller. And because we have variations in the way that we do transcription, variations in RNA processing, and variations in translation, and then after translation, we end up with a hell of a lot more proteins at the end of it. And that is a difference between the genome and the proteome.